doctor?
Well, Wolf, I'm sorry to tell you, but it seems to be there's a little bit of a problem with your health. Let me show you. Well, see, Wolf, your problem seems to be in your interphase of the cell cycle. See, the interphase is 90% of the cell's life. It starts with the G1 stage. What happens here is the cell increases in size and doubles the amount of organelles, specifically mitochondria and ribosomes. This becomes like the new cell. This stage comes right after the cell is divided in the previous cycle. Here, it accumulates the materials that are needed for DNA synthesis. After that comes the S stage. The S stage is also called the synthesis stage. The goal is to create two identical DNA double helixes that will form this chromosome. Next comes the G2 stage. Here, proteins are synthesized that assist in the cell's division. The problem that you seem to be having is you are skipping the G2 stage and going straight to mitosis. This is common with many forms of cancer, Wolf.
Is there any cure?
The treatment for this disease is going to be very expensive. I hope you know that.
What are you doing, Tank?
Hey, Wolf. I was just investigating this cell division raid. Look at this. I got all my guys working on the same case. It's crazy. Yeah, there's a lot of money in cell dividing.
Interesting. That's interesting.
Giuseppe,
Oh,
it's your former teacher,
no.
Mr. Tight.
Hey.
Listen, I hear you're in the business of making cell trade.
Whoa, no.
Listen,
no,
I'm not
that's-
going to turn you in. I need your help.
What? With what?
I want to make cells.
Well, I could help you sell them, but I don't know how to make them.
I can take care of that.
In mitosis, we have the first stage of mitosis is prophase. In early prophase, the nucleus has just started to fragment. Later in prophase, the nucleus disappears and the chromosomes become visible. Also, the centrosomes start to move apart. Next is prometaphase. The connected core of each chromatid is attached to a spindle fiber. Metaphase. Centromeres of the duplicated chromosomes align at metaphase plate in the middle. And then anaphase. The sister chromatids part and become daughter chromosomes that move towards the spindle poles. In this way, each pole receives the same number and kinds of chromosomes as the parent cell. The last stage of mitosis is telophase. This is where the daughter cells are forming as nuclear envelopes and nucleoli reappear. Chromosomes will become indistinct chromatid. Do you understand, Giuseppe?
Okay, Mr. Tight, that makes sense, but we gotta make sure that we make animal cells and not plant cells. Yo, this is cytokinesis in an animal cell. In an animal cell, a cleavage furrow begins when anaphase is closing. A band of actin filaments called the contractile ring pulls around the cleavage furrow, tightening it. This continues until the two cells are separate. Yo, in plant cells, the cell walls don't permit separation. Instead, a small flattened disc appears between the two cells. The cell plate is just newly formed plasma membrane that expands outward until it fuses with the old one. Yo, Mr. Tight, let's divide some cells.
Giuseppe, listen to me very carefully. Take an Erlenmeyer flask, a beaker, a 100 milliliter beaker, and 2 pipettes. Okay? This is the finished product. This is the purest cell matter I've ever seen.
That could sell for big bucks, Mr. Tight. Yo, Dad, what's for breakfast?
Science.